and my research is on visualization of the evolution of a virus genetic sequence, which is a design study in the intersection of biology and visualization. And the tool that I've developed is for the domain experts to explore and make sense of a big data set. So this is my research process that I've translated the data set from the original domain into data types and tasks of InfoVis domain. And then I've designed visual encodings and interactions to support those translated tasks. I've used InfoVis design guidelines during the whole process, and also my dataset is a benchmark dataset, and hopefully I assume that it's based on a deep understanding from the domain. So a bit about the domain vocabulary. A gene sequence consists of single nucleotides, which is coded as A, C, T, and G. A virus cannot replicate without using a living cell's machinery. That's why virus attacks a cell, makes copies of itself, and growing its population among the same person, and eventually spread the disease to others. But during this viral replication, the whole gene sequence has to be copied. But because nothing is perfect, typically some mistakes happens. And as a result, you have mutations of virus strains. And my tool is to help analysts to connect these changes to having different characteristics of a disease during for different patients. So the data comes in two tables. The first table, the first row, has the information about the original virus, and all the rows have information for each of the patients. A column is position in aligned sequences, and cell attribute is a nominal data with the dynamic range of A, C, T, and G. And the second table is about disease characteristics. So a column is the characteristics, and a cell attribute is ordinal data, which has a dynamic range which is different for different characteristics. For example, for symptoms it could be mild, severe, and moderate. So this is how I translated the dataset into using InfoVis vocabulary. I've defined a derived cell variable.
I'm sorry. Yeah.
So this is how I translated the data sets using InfoVis vocabulary. So I've defined a derived cell variable, which is I've kept the first row, the original sequence, but for the other cells, I've defined whether or not it is changed in comparison with the original sequence. So if there is no change, there would be 0. If there is a change, there would be 1. And for the second table, I've defined a derived variable, which is an average of all the disease characteristics for each patient, because analysts, they want to have the overall danger level for characteristics. Okay, so this is shown how I have translated the data set. Okay. And this is a visual encoding which is universally used for the first table, and I just picked the colors here, and I've also justified the design, which is here they use hue as an extremely powerful channel for categorical data. I've picked purple for not change category and yellow for change category. And I've printed this in black and white, and I've also checked that in this check website for avoiding color blindness, and it works. So here is important, the important thing is that whenever I want to design, I would look at the order channels based on effectiveness of them for different types of data. For example, hue is very powerful for categorical data. Or for example, position could be very powerful for ordinal data. Okay. The domain task is identifying substitutions that lead to an increase in different disease characteristics severity. And this is an output for particular characteristics. This is an example. For example, G changes to E in this position, T changes to A in another position. They are equally important, and they are the most important changes, and then after that. So I translated this using InfoVis vocabulary. This is find columns that lead to the most severe rows, and to do that, I need to sort rows according to the value of a certain characteristics, and then filtering columns until I find the answer. So this is a translation from the original task to InfoVis task. This is the whole design for that task. It has the matrix view, it has a table lens view. I've used position and redundantly saturations of different colors for encoding ordinal data. Also, I have an overview, and the user can define different groups with different columns. I've used a length channel for a measurement for a particular measurement for a column, and this graph is showing correlationship between different columns. I've already implemented that in processing language, and I'm doing exploratory design with domain expert and refining the idea iteratively, and hopefully it will be a module and it will be used in a more general software or framework. A gene sequence consists of single nucleotides, which is coded as A, C, T, and G. A virus cannot replicate without using a living cell's machinery. That's why virus attacks a cell, makes copies of itself, and growing its population among the same person, and eventually spread the disease to others. But during this viral replication, the whole gene sequence has to be copied. But because nothing is perfect, typically some mistakes happens. And as a result, you have mutations of virus strains.